Welcome to Biopharm International's podcast, monitoring and controlling dCO2 in cell culture and fermentation. This podcast is brought to you by METTLER TOLEDO Process Analytics, who manufacture real-time analyzers for pharmaceutical manufacturing and process control. To find out more, please go to www.mt.com. And now, here's your host for this podcast, the contributing editor for Biopharm International, Cindy Dubin.
Hello, everyone. This is Cindy Dubin, contributing editor for Biopharm International, and I'm here with Olivier R. Berteau, Pharmaceutical and Biotech Industry Manager at METTLER TOLEDO Process Analytics. Thank you for being here today, Olivier.
Thank you for having me. It's a pleasure to be here.
So to get started, we know that accumulation of dCO2 as a critical process parameter in culture media is often ignored in fermentation and cell culture. And I wanted to ask you why you think this is a missed opportunity for process control.
Indeed, CO2 is a gas, and it seems logical to monitor it as a gas. But in fermentation and in cell culture, the dissolution and dissipation of CO2 is a very dynamic phenomenon, hence the importance of measuring it in its dissolved form, because that's the form in which it directly affects the cell population.
So more specifically then for cell culture, what makes dCO2 critical to measure at any scale?
Well, it's a complex topic, but it all comes down to control of the process. The carbonate buffering system used with the cell culture media, the accuracy of the measurement of the pH, and the dCO2 levels combine to allow you to monitor and control the pH at a very fine level, and therefore limit, for example, the excessive accumulation of lactate and ammonium in the cell culture media. In fact, as the inline measurement of dCO2 provides the same data as an offline BGA, it is easier to compare CO2 data. And the precision obtained for monitoring and control is very easy to qualify. And since METTLER Toledo pH sensor for cell culture have almost zero drift post-sterilization, this correlation between pH and dCO2 is of great interest for cell culture.
So, just so I understand correctly, are you saying the dCO2 levels have an effect on pH control?
Yes, and there are several publications that explain how it could positively impact pH control. For example, when using buffered media at the defined pH and dCO2 content, the addition of bicarbonated pH solutions can be reduced and potentially eliminated because dCO2 content is monitored and pH becomes more stable and smooth to control.
Okay, thank you. So, going back to fermentation, what makes dCO2 critical to monitor?
Basically, as long as dissolved CO2 is able to pass both ways a cell membrane, looking at dissolved CO2 in fermentation and monitoring its accumulation makes sense. Many engineers are measuring CO2 as a gas in the off-gas or the overlay of the fermenter. Sometimes multiplexing the measurement is applied, complicating and inducing a delay in the monitoring. With dCO2 measurement, the content can be monitored in real time in the media. For example, a citric acid fermentation using dCO2 monitoring and thresholds help to maintain batch-to-batch productivity expected. Dissolved CO2 is not always critical with microbial systems, but as I've observed, it should be implemented more often in various fermentation processes. And because dissolved CO2 is not only used to control pH in fermentation, the dissolved CO2 measurement is a direct output of the fermentation.
Interesting. So, a fermentation could be monitored as input and output data that are used to influence the fermentation balance?
Correct. Looking at a fermentation process as inputs and outputs dCO2 in fermentation could help to monitor, for example, the addition of a carbon source, an expression, and maintain the optimum dissolved CO2 required by cell population.
So, Oliver, could the same be simply applied to a cell culture process?
Yes, but for mammalian or insect cells, manipulating pH and CO2 could become more valuable than helping to better monitor the addition of a carbon source. Don't get me wrong, dissolved CO2 will impact media osmolality, growth rate, and quality attributes of a protein or a monoclonal antibody. So like a CO2 incubator, the possibility to maintain dissolved CO2 at a fixed set point in media, or knowing the dissolved CO2 accumulation is dynamic, it's also interesting to maintain different dCO2 profile or set points at specific time during a batch.
How would you recommend that someone measure dCO2 in cell culture or fermentation?
Well, METTLER Toledo has a dCO2 sensor specifically designed for bioprocessing. It's accurate, used in commercial phase since years by our client under fed batch or perfusion, with runs exceeding sometimes 300 days. So the technology is robust and qualified. The accuracy, simplicity of calibration, and performances are proven for bioprocessing, and it will help to get the measurement you need in cell culture fermentation at all scales.
Wonderful. Thank you for this informative overview, Oliver. We truly appreciate you being here today.
Thank you for having me.
This has been Cindy Dubin, contributing editor for Biopharm International. Thanks to all for listening.
You've been listening to Biopharm International's podcast, Monitoring and Controlling dCO2 in Cell Culture and Fermentation. This podcast is brought to you by METTLER TOLEDO Process Analytics, who manufacture real-time analyzers for pharmaceutical manufacturing and process control. To find out more, please go to www.mt.com.